think I will take it away now. It's my great pleasure and honor to introduce today's speaker, Dr. Sonja Best. She's visiting from the Rocky Mountain Laboratories in Montana, and she's currently the Deputy Chief of the Laboratory of Virology at Rocky Mountain Laboratories, and that is part of the NIAID. And her research group is really focused on understanding the host-pathogen interface associated with intrinsic antiviral responses of cells to emerging RNA viruses. So the Rocky Mountain Laboratories is really our closest BSL-4 laboratory from here, and they have all the biosafety laboratories there and can really go and delve into the most dangerous viruses that are around. Dr. Best herself earned her PhD from the Australian National University in Australia, where she has initially worked on myxoma viruses, but she then joined the Rocky Mountain Laboratories, where she conducted her postdoctoral research and then eventually became an independent lab head herself. She received tenure and was promoted to Senior Investigator in 2017. She has been recognized with many awards and keynote lectures. She has been honored with an NIH Director's Award and a Merit Award, and she's also on the editorial board of several important virology journals, Journal of Virology, PLoS Pathogens, and Science Translational Medicine. And her lab really works on a broad spectrum of different viruses, ranging from myxoma to Flaviviruses, but also very interesting mink viruses. But her focus is really on the innate immune response and how cells respond to these different viruses. And today she will focus our attention on the mitochondrial response here in the interaction with Flaviviruses. So without further ado, we're extremely delighted to have her. Thank you so much for coming, and we're looking forward to your talk.
There we go. Thank you very much. Thank you, Melanie. And I hear the students for this very, very kind, or the trainees for this very, very kind invitation, and I wish I was certainly there in person, mainly because of the people, but also because of the weather differential here in Montana. So today I'm going to tell you about our work with Flaviviruses. As Melanie said, we have the BSL-3 and BSL-4 labs here, and so as we've seen a number of different emergent events over the last decade or so, we do have research programs in Ebola virus, of course, SARS-CoV-2, but the Flaviviruses is the group that keep drawing me back in. I just think they're so fascinating. And of course, in terms of, the slides to advance, in terms of their global everyday impact on human health, it's quite substantial. Of the 53 recognized species of Flaviviruses, 40 of them are recognized to have pathogenic potential for humans. And of course, they have demonstrated their ability to emerge and re-emerge on grand scales. I was actually just home in Australia just in March, and Japanese encephalitis virus, that we traditionally consider a disease of Southeast Asia and the tropics, was emerging down here in very temperate zones in Australia. So they're not confined to traditional geographical locations any longer. And probably one of the most explosive examples of this was Zika virus that emerged into South America in 2014. And although we studied most of these viruses to compare and contrast their different strategies for interactions with host cells, I'm going to concentrate mainly on Zika today. So, as I said, the success of these viruses as emerging pathogens is really because they are arthropod-borne, transmitted by the bite of an infected tick or mosquito. And then you have initial replication of the virus in the skin, predominantly in keratinocytes. And then DCs are also a target cell. The virus very quickly, probably from minutes to hours, can then translocate to draining lymph nodes where it is amplified, and you eventually have establishment of this primary viremia where virus is present both in cell associated with monocytes as well as free in the circulation. So for most people, this may be an asymptomatic infection and can be controlled at this point, or it might be associated with a febrile illness or rash. But in a subset of people, these viruses are then able to cross critical barriers. And this includes the central nervous system for some of these viruses that are well known to us, including West Nile virus, as well as some that people may not be as familiar with, such as tick-borne encephalitis virus or Japanese encephalitis virus. When Zika emerged, it also had this propensity to affect the central nervous system, but this time it is in the developing brain of the fetus when an expectant mother is infected. Or there are other viruses that are really quite fascinating in terms of the inflammatory dynamics, including dengue and yellow fever, that tend to be more viscerotropic. Something that they seem to have in common is at least biomarkers of the inflammatory response. So where this has been characterized in people, predominantly with Zika and dengue from Eva Harris's group, but also other groups, there seems to be this core set of chemokines as well as type 1 interferons that are readily detectable in the sera of infected patients, with increased expression of these various chemokines associated with poorer outcomes of infection. We'll come back to that as we go through this talk. So these, you've probably seen a lot of this due to Melanie's lovely work, but these flaviviruses are thought to have relatively simple replication strategies, largely because after they enter the cells, the genome is really just a single-stranded positive sense RNA that is then rapidly translated on the endoplasmic reticulum. And as it's translated, it causes all these membrane rearrangements that really form the basis of these replication complexes. And then eventually, as the structural proteins are made, they will associate with the RNA and bud into the ER. And then those immature virions will transit through the trans-Golgi system, maturing by enzymatic cleavage as they go to release infectious virus. But it's not quite so simple. These viruses cause massive rearrangement of the endomembrane system. And these are just some examples taken from a paper from Ralph Bartenschlager's group, where you can see this extraordinary membrane reorganization. This occurs kind of in two general phenotypes. You have invagination of the ER here, and these, they're often termed peas in a pod, but these replication vesicles where RNA replication occurs. And then you also have this kind of second form, which is called these convoluted membranes, which is contiguous with these virus-induced vesicles, but the exact functions of these kind of convoluted membranes are not so apparent, at least to me. And as I said, the reason for this massive rearrangement is because as this RNA gets translated, you have the this single polyprotein has many transmembrane domains, and as it inserts itself into the membrane of the ER, particularly these small hydrophobic proteins are responsible for all these different membrane curvatures and proliferation. In addition to this, there are two proteins shown here, the NS3 protein, which encodes the viral protease, as well as the NS5 protein, which encodes the RNA-dependent RNA polymerase that are responsible for really driving RNA replication. And my great interest over the last more than a decade has been this non-structural protein 5 in its roles in interferon antagonism. And it's the study of this NS5 protein that's kind of led us to the story that I'm going to tell you about today. So when we think about the mitochondria in the course of flavivirus infection, we really think about it as a platform for organizing the host response to infection. So in the context of flavies, we know that both TLRs and RLRs are important, but particularly for the RLRs, RIG-I and MDA5, they're going to signal through this single critical adapter protein called MAVS, or mitochondrial antiviral signaling protein, that is constitutively associated with the mitochondria to then activate these downstream signaling cascades, resulting in production of the interferons and then signaling through the JAK-STAT signaling pathway. And as many of you know, NS5 from these various flaviviruses has been shown predominantly by our group and that of Adolfo Garcia Sastre to suppress various signaling intermediates in this side of the pathway. The ability of these viruses to suppress this kind of the initiation or production of interferon, it tends to be very different between different viruses. So when we were looking at NS5 function, in terms of actually antagonism of interferon signaling, we have conducted a number of different proteomic studies, mainly yeast-2 hybrid studies or screens, trying to find various interactors and then link them back to aspects of the innate immune response. And so the story that I'm going to tell you about today that led us to mitochondria was an putative interaction between NS5 from Zika virus and this host protein called a Juba virus. And this project has been carried out by Sanket Ponya, a postdoc in the lab, with very important contributions from these 4 lab members here, particularly Shelley and Kristen. So Juba has been characterized in the past in terms of its role as a scaffolding protein with either roles in mitosis, which we'll come back to a little bit, and cellular transformation. It belongs to this larger family of about 5 different proteins, which have a pre-limb domain and then these limb domains, which are just interacting scaffolds where they have been known to really, well, a primary role, at least of a Juba, is the relief of auto-inhibition of serine threonine kinases, particularly those involved in mitosis. So here you can see, this is just a reminder for me that a Juba has roles in the relief of inhibition of kinases such as Aurora A as well as CDK1. So in the absence of a Juba, the exit or the ability of cells to undergo mitotic exit is reduced. But as we always do when we find these interactions, we tend to take that protein of interest and either knock it out or overexpress it, and then stimulate cells with various agonists of innate immunity to see if we can link these proteins with the innate immune response with the, I guess, viral-centric or innate-centric view that these viruses really have to replicate their RNA and escape innate immunity, and that's the 2 functions of most of these proteins. So with this in mind, what we did here was just to overexpress MAVS as the single adapter protein that is going to then, overexpression of that protein alone is sufficient to get aggregation on the mitochondria and drive downstream production of type 1 interferon. So here you can see our relative levels of interferon as measured by real-time PCR. But if we co-express MAVS here together with a Juba or its related family member, LIMD1, we get this very marked reduction in MABs-dependent type 1 interferon expression. And this is associated at the level of these Western blots, at least, with an overall reduction in the level of MABs expression. And so at this stage, we're thinking, this is wonderful. We've got a new kind of key to RLR signaling that these viruses might target to suppress signaling. And so we went further to look to whether or not this was kind of a generalized feature of ajuba. And so here you can see we've just overexpressed ajuba here in green in cells labeled with TOM20 for the mitochondria, and then infected those cells with Sendai virus as a kind of canonical driver of this RLR signaling pathway. And you can see either in these IFAs or by these intensity plots here that ajuba seems to be somewhat localized with mitochondria, even in resting cells. But if you stimulate with Sendai virus, now you have this really quite marked recruitment of ajuba to the mitochondria. But unfortunately, this is where the story became difficult for us. And it's because we tried all these different immunoprecipitations and other ways to try and work out where in this RLR signaling pathway ajuba could be interacting with. And we didn't find any data really that was convincing. And so we instead tried to concentrate on this degradation event to see if that would give us some clues as to what role ajuba might be having. And so here we did the exact same experiment where we're overexpressing MABs together with ajuba or its cousin LIMD1. And we've treated cells with baflomycin A1 to inhibit the lysosome or epoxamycin to inhibit the proteasome. And sure enough, both pathways seem to recover this overall loss of MABs protein expression in the presence of ajuba. But when we kind of took a step back and started putting in some additional controls, which is always helpful, we actually put in this marker TIM44 for the outer membrane of mitochondria as a control. And you can see that it wasn't just MABs that we were losing, but also these outer membrane markers that are also responsive then to inhibition of both of these degradation pathways, lysosome and proteasome. So there's this raised the possibility to us that it's really not the MAB signaling pathway that's being targeted, but perhaps mitochondria more generally. And this process is where mitochondria turned over also has a canonical pathway called the PINK1 Parkin pathway, which I'll come back to in detail in the very next slide. But to try and determine if this was actually responsible for our phenomena, we took HeLa cells, which HeLa cells are naturally very low in Parkin. And so they have compromised pathways of mitophagy. And we overexpressed MABs here together with ajuba. And you can see that we now we've lost our effect of ajuba-mediated degradation of MABs. But if we reintroduce wild type Parkin, but not an active site mutant of Parkin here, we can recover this degradation phenotype. So it suggests that we didn't have anything controlling the RLR signaling pathway, but now we may have unwittingly kind of uncovered a new aspect of response to damaged mitochondria, which is also going to occur in terms of mitochondrial membrane depolarization when you overexpress MABs. And so to look at this more generally, we expressed ajuba in cells and then just induced widespread mitochondrial depolarization through the addition of this CCCP, which is a cyanide derivative, and fractionated those cells into cytosolic fractions or fractions containing the mitochondrial or membranes in general, but will contain the mitochondrial fraction as noted by this purification of TIM44 again. And you can see that within an hour of mitochondrial depolarization, you have marked recruitment of ajuba to those mitochondria. So it's probably not got anything to do with MABs per se, but just the damage to mitochondria that you're inducing when you activate these pathways. And so, as I said, the main pathway that is responsible for the clearance of damaged mitochondria, the selective removal of damaged mitochondria, is the PINK1-PARKIN pathway. These proteins are both mutated in particularly juvenile onset Parkinson's disease, so they're of high interest in understanding these other pathways in neurodegeneration. But the pathway itself is really this very elegant way that your cells are constantly sampling the health of your mitochondria. So here we have PINK1 that is constitutively expressed, and the little tail gets imported into the inner membrane of the mitochondria dependent on this mitochondrial membrane potential being intact. Here, this tail gets cleaved by inner membrane proteases, resulting in the release of PINK1 and its constitutive degradation. However, when mitochondria become depolarized and you have this loss of membrane potential, the tail of PINK1 cannot be imported, so it cannot be cleaved. And now you have this local accumulation of PINK1. PINK1 is a serine-threonine kinase that's present in an autoinhibited state, but the accumulation of PINK1 is thought to result in transphosphorylation of PINK1, resulting in its activation. PINK1 can then phosphorylate PARKIN, which is an E3 ubiquitin ligase, also present in an autoinhibited state in the cytosol. This results in its activation. And then PINK1 and PARKIN cooperate together to build these ubiquitin chains that are uniquely modified by this phosphorylation, which we'll also come back to, on the outer membrane of, on proteins in the outer membrane of the mitochondria. And these are the chains that are ultimately responsible for recruiting the selective mitophagy receptors that then build the autophagosomal membrane that eventually then fuses with the lysosomes to result in mitochondrial clearance. And it's the reason that you can recover intermediates of this pathway by inhibition of both the lysosome is because of this end stage of mitophagy, as well as the proteasome is generally because PARKIN functioning as an E3 ubiquitin ligase is resulting in proteasomal mediated degradation of these outer membrane proteins. So this all fits that this loss of MABs that we initially looked at is really, we're not looking at MABs per se, we're looking at this mechanism of mitochondrial clearance following a damaging event. So just before we get on to why the virus might be wanting to do this, we further wanted to define the role of ajuba in this pathway and whether or not it was just, is it actually actively participating in these signaling events? And so to do this, we generated ajuba knockout mice and made MEFs from those mice. And then we labeled those mitochondria simultaneously with mitotracker red and mitotracker green. And the green fluorescence is going to be sensitive to quenching in the lysosome. So when you induce mitochondrial depolarization with this CCCP again, you're going to get a shift in the red to green ratio as the green signal gets quenched. And you can see that in the absence of ajuba that this event is compromised, and it's actually not that dissimilar than if you inhibit the lysosome in its entirety with baflomycin A1. So we also looked at this phosphoserine 65 ubiquitination event. So this phosphoserine 65 modification of ubiquitin is highly diagnostic of PINK1 activity, or highly unique to PINK1 activity. And you can see that in the presence of overexpressed ajuba that we're not triggering the pathway by itself, but we do need to add this further depolarizing stimulus to see the phosphoserine 65 ubiquitin, which is increased in the presence of ajuba. And so we wanted to know, as I said, that PINK1 is a serine-threonine kinase that hangs around in the cell in terms of this in an autoinhibited state, and you can see here that what we did was we expressed PINK1 or a juba in E. coli. So this is completely in vitro, incubated them together, and you can see through these reciprocal immunoprecipitations that they certainly are able to bind to each other in the absence of any other proteins. And then if we incubate a juba with, or increasing amounts of a juba with a constant amount of PINK1, with this protein level all normalized by the presence of BSA, you can see that if you stain these Western blots for the phosphothreonine 257, which is a specific modification, auto phosphorylation event of PINK1, that this is increasing with increasing nanomolar of a juba, and actually when you get to about a 1-to-1 ratio, you have optimal autophosphorylation of PINK1, and this is quantified here. So it seems that there could be a function for a juba in its more canonical role in terms of the relief of kinase autoinhibition, which essentially enables a highly controlled but accelerated activation of these kinases. So in summary, it's really what I just said, in trying to work out this role of a juba, it seems to be responsive to mitochondrial depolarization, where it is being recruited to mitochondria. We think this recruitment is dependent on the stabilization of PINK1 at the mitochondria, and then it is functioning to accelerate activation of PINK1 and also thereby accelerate activation of Pkr to build these phosphoubiquitin chains. And so this then resulted in the hypothesis for us that NS5, the association of NS5 with the juba, is really going to be to drive mitophagy with the idea that all these viruses want to do is basically avoid RLR MABs signaling. And actually a number of viruses, including hepatitis C, for Melanie's work, have been shown to increase the turnover of mitochondria, in part specifically to avoid these signaling events. But what we found was quite surprising and a little confusing for some time, is that these, at least for Zika virus, that the interaction with NS5 and a juba is to suppress mitophagy and actively retain these damaged mitochondria in cells. And I'll show you some data for that. And in that way, it's actually, I think these are the flaviviruses are the first viruses to have been described to suppress these mitophagy pathways. So just to show you a little bit of evidence to support my claim, we looked at this with Zika virus infection in cells. And the way that we tried to do this originally was using again this HeLa system. So as I said, HeLa systems are naturally low in Pkr, but they've been used to basically reconstitute Pkr or to eliminate PINK1 so that you can look at the specificity of these signaling events. So we took those cells, transfected them with a construct expressing ubiquitin with a HA tag, and then you treat the cells with CCCP to depolarize the mitochondrial membranes, and then we've actually stained for that phosphoserine 65 modification that's so specific to PINK1, and then you can kind of enumerate these puncta as a measure of mitophagy specifically in infected cells. And so when we couple this with staining for the virus, you can see that not only does Zika infection not induce an increase in this signal, but when you then treat cells with CCCP, infected cells with CCCP, it really is able to impart a quite remarkable block to this signal. So consistent with the juba being required for phosphoserine 65 ubiquitin accumulation, Zika is able to suppress the pathway at that precise point. So is this attributable to NS5 function? Is kind of the next question that we asked. And you can see here that by immunofluorescence, an example of what we think NS5 is doing. So what we did here was we expressed a juba by itself here in red. TOM20 is in gray, and so you have this kind of nondescript puncture of a juba, often associated with mitochondria again. We've overexpressed MAVs, and MAVs, that gives us two things. Number one, it gives us very specific localization of the mitochondria here in gray, as well as we know that these mitochondria will be selectively depolarized. And then we've expressed Zika NS5, which localizes predominantly to the nucleus, which is a little counterintuitive, but you can, when you overexpose these cells, you can see that there is some NS5 in the cytosol. And certainly when you fractionate cells and followed by Western blot, you can see that NS5 is present in the cytosol. So here we have an example of the fluorescence where we've just expressed MAVs. Sometimes it's MAVs is expressed by itself, as in this cell, or together with a juba, as in this cell. And you can see that a juba here in red is kind of interweaving itself around these mitochondria, and you are actually losing your aggregates of MAVs consistent with this degradation event of damaged mitochondria. However, when you introduce Zika NS5, and now this is overexposed so that you can see some of this Zika NS5 is associating with a juba in a MAVs positive cell, you can see that a juba is really, doesn't even really see these MAVs positive mitochondria. So it's kept away from those depolarized mitochondria. And if you do Western blots of your whole cell culture, you can see that a juba is not recruited to the mitochondria, and neither is Parkin. So it seems to prevent those downstream signaling events. So the implication from this is that these viruses, or at least Zika virus, is actively retaining damaged and depolarized mitochondria in cells. But what is the consequence of that to virus replication and to pathogenesis? Is there a consequence? So to look at this, we turned back to our MEFs, so these mouse embryonic fibroblasts from wild type mice or a juba knockout mice, or we've got pink one knockout mice in some subsequent slides as a control. And you can see just from these EM images that in the juba knockout MEFs, it was really quite easy to pick up evidence of mitochondrial damage in these cells. So you can see these kind of dysregulated membrane structures. Sometimes you can see differences in the cristae structures. And if you then infect these cells with Zika virus, so in yellow, I have noted the virus-induced vesicles that you can see in these infected cells. But these, in the absence of a juba, you see really quite widespread damage of these mitochondria. Some of them aren't even really recognizable as mitochondria. And you also see now this accumulation of convoluted membranes here, often kind of bordered by these extended or elongated mitochondria, which is a concept that was first raised again by Ralph Bartenschlager's group in 2016, that they were talking more about the fusion of mitochondria, which is a separate event that I won't go into, but that these elongated mitochondria were somehow supporting the formation of the convoluted membranes. And we actually also see that specifically in these juba knockout MEFs as well. So we think that these mitochondria are supporting some of this membrane proliferation. And I'll slightly touch on that as we don't have all the answers actually for this, but I will touch on that a little bit more as we go through. So So what are the potential consequences of suppressed mitophagy and keeping these damaged mitochondria around in cells? For the virus, there's probably a lot of benefits. So we know that these mitochondria are going to be producing reactive oxygen species. There are a number of viruses that actually thrive on ROS, including Flavivirus are thought to, where these reactive oxygen species can actually aid enzymatic function for the replicative proteins. At least through the process of oxidative phosphorylation, you could have increased local concentrations of ATP. Mitochondria are critical, and particularly the communication between mitochondria and other organelles and the organelle contact sites, particularly the ER, as well as peroxisomes. They're very important for changes in lipid metabolism. As well as then, when you retain these damaged mitochondria in cells, this is actually a major source of amplification of the integrated stress response, which I thought was really just the ER, but it has contributions from mitochondria as well. And this is, viruses do this to result in phosphorylation of EIF2 alpha and inhibition of cap-dependent translation of cellular proteins, which is a hallmark for Flavivirus replication. But you can't do this without the cell noticing that the mitochondria have gone awry. And this is because the mitochondria start to look like the ancient bacteria from which they have evolved, the protobacteria from which are the origins of our cells, right? Or complexity of our cells, at least. And so in this case, these mitochondria, of course, are going to start pumping out ROS and exposure of cardiolipin that can both result in inflammasome activation. And in particular, you also have release of nucleic acids, the mitochondrial DNA, that's going to result in cGAS sting activation, as well as mitochondrial RNA that's known to be detected by protein kinase R and MDA5. And importantly, these two events, what the virus wants and what the host sees, are linked through a couple of different mechanisms, but the mechanism that I'm going to concentrate on is really this amplification of the integrated stress response and inhibition of translation. Because as for EIF2 alpha to get phosphorylated, you have to have activation of one or more of the 4 upstream kinases, of which protein kinase R is one of them, resulting in ATF4 expression or translation and kind of expression of these proteins that would ameliorate the stress response or ameliorate the stress. So we know that this stress response is triggered by flaviviruses, but is the retention of damaged mitochondria in cells contributing to this integrated stress response? And so we can see very clear evidence that this is in fact the case in our Juba knockout MEFs here. And we've got the pink 1 MEFs now as a control. And you can see that as this main readout for the stress response of ATF4 translation, we kind of barely see it in our wild type MEFs. It's amplified in the absence of pink 1. And actually in the absence of a Juba, not only do you have this marked translation of ATF4, but it seems to be sustained throughout this time course of 72 hours that we examined this over. And in terms of looking at a potential role for PKR, not only is its phosphorylation increased in the absence of a Juba compared to wild type cells, but now you can see that in the absence of a Juba or the absence of pink 1, you have marked basal phosphorylation of PKR, which is quantified here in these 24-hour time series. And so we definitely have both activation of PKR and sustained activation of the integrated stress response. And so whether or not these are important to replication is something that we did through use of inhibitors. So ISRIB here is going to inhibit EIF2 alpha phosphorylation. C16 is an inhibitor of PKR. And we've shown in the paper that we can suppress the activation of these two molecules. And you can see that not at early times post-infection, so we've had the inhibitor on for 24 hours at each case, at each time point, but so not at these early time points, but certainly by 48 and 72 hours, we get about a log reduction in the release of infectious Zika virus with these two inhibitors. So the integrated stress response, this is already known, contributes to favorable replication conditions, and presumably this is associated with some of those membranous changes that I've alluded to. But what's the driver of PKR then? When we think about protein kinase R, it's very classically studied as an ISG that recognizes viral double-stranded RNA. But as I noted that we had marked activation of PKR even in our, just in uninfected cells if Ajuba or PINK1 were knocked out. And so we looked at a potential role for mitochondrial RNA in actually driving this. And interestingly, if you use that J2 antibody that we as virologists always use to pick up our replicating virus in infected cells by fluorescence, a little known fact is that in uninfected cells, 95% of what J2 recognizes is actually mitochondrial RNA. And so we could use this in a fluorescence assay and then quantify the signal intensity for that double-stranded RNA staining in cells. And in the absence of PINK1 and Ajuba, you can see that we have marked increases in this intensity of J2 staining, so mitochondrial RNA. And you can deplete mitochondrial RNA by treatment of cells with low levels of ethidium bromide for an extended period. And so we've done that here in our various MEFs and verified that it's having the effect that we think, because we've lost expression of cytochrome B, which is encoded by the mitochondrial genome. But another mitochondrial protein, MFN1 or mitofusion1, which is encoded by the nuclear genome, is unaffected. And so when we do this in the context of Zika virus infection, you can see that depletion of that mitochondrial RNA now results in suppression of activation of PKR during infection. So I'm not saying that there's not a contribution of viral RNA, but there is clearly a very significant contribution of mitochondrial RNA or self-nucleic acids in driving this PKR activation and amplifying the integrated stress response. And that is ultimately fueling, I think, these membranes and some of the later stages, which I don't fully understand, but later stages of viral replication. So does this have any implications for pathogenesis or these inflammatory responses that are really the biomarkers of a flavivirus infection in humans? And so to look at this, we performed RNA-seq on our MEFs, infected or not, with Zika virus. And you can see just from the differentially induced genes here, or expressed genes, that Ajuba and PINK1 have the vast majority of their changes in gene expression are linked, suggesting again that these two proteins are functioning in the same biochemical pathway. But in terms of inflammatory responses, and we could definitely see the ISR in these gene signatures, but in terms of the inflammatory response, the most predominant genes that are kind of clustered up here are these chemokines, as well as the type 1 interferons, as being increased in the absence of mitophagy, effective mitophagy. And so we looked at this to see whether or not this really is the mitochondrial RNA and PKR activation that are driving the chemokine responses. And indeed, we think it is. So here we have our experiment again. This, I should have said, this was in primary human fibroblasts, so where the mitophagy pathways are now intact. And when we looked at these chemokines, and of course, you can see them being expressed in our 24-hour increments of the time course that we looked at following Zika virus infection. If we inhibit ISRIB, or if we use the inhibitor ISRIB, where we're inhibiting EIF2 alpha phosphorylation and suppressing virus replication by a battle log, we not only don't ameliorate these various chemokines, but they're often slightly exacerbated. And this is consistent with the fact that if we're inhibiting at this level, we're not actually allowing this integrated stress response to help to resolve the stress that these cells are under. However, if we use our inhibitor of PKR, that is, so this C16 inhibitor, which has an pretty equal effect on virus replication, only now are we able to get rid of or reduce the expression of these various chemokines, suggesting that it's actually the mitochondrial RNA that's most likely driving these inflammatory responses, because presumably the replicative intermediates through at least this first 48 hours are largely similar. So now we can uncouple inflammation from really the replicative or the RNA, the viral replicative intermediates, and start to implicate, I think, some of this mitochondrial RNA as driving inflammation. So finally, in the last couple of minutes, I'm just going to shift this to our in vivo studies to see if, so can we see signatures of this integrated stress response or these chemokine responses in animals, and does that really have any effect on viral pathogenesis? And so this is just RNA scope using probes that are specific for the Ajuba mRNA. So you can see that Ajuba expression is enriched, particularly in epithelial cells, so in keratinocytes here in the skin, as well as in Sertoli cells in the testes, so two key targets of Zika virus replication. But we also see expression in other epithelial and endothelial targets, as well as, to a lesser extent, but certainly present in neurons. And then it was really screaming in the liver and hepatocytes and endothelial cells. So all very key targets for various flaviviruses. And so we took our knockout mice here and infected them with Zika virus. I must say that we did this in presence of the anti-IFNA blocking antibody, so that we could get replication of Zika virus at all in these mice in the presence of a mouse interferon response, which the virus can't counter or effectively counter. And so you can see then what we did was just measured chemokines and various cytokines here in the serum at 3 days post-infection or 5 days post-infection. So in the blue is our Ajuba uninfected or infected Ajuba knockout mice compared to their black 6 controls. And what was quite remarkable was that at 3 days post-infection, we could see these exact signatures of these chemokines. But if you looked at something like interferon gamma or something like that, these cytokines weren't affected. It was very specific to the chemokines. But our responses that we saw that were augmented in the absence of Ajuba, those differences were largely lost at 5 days post-infection. So the virus seems to be able to do this effect itself, or maybe the later stages are Ajuba independent, but we could see this signature early. And when we looked at this in terms of virus burden, we saw again quite remarkably that in those Ajuba knockout mice, we now had an 80-fold greater virus burden, and this is infectious virus in the spleen at this 3-day post-infection, or 16-fold greater in the brain. And this, interestingly, was not due to an intrinsic advantage of Zika virus to replicate, because these peak titers at 5 days post-infection in these critical organs were not different from each other. And in addition, the amount of infectious virus present in the serum at this critical time point of 3 days post-infection was not different. And so this has led me to tentatively conclude, that these responses are not only amplifying inflammation, but it's something to do with either those mitochondrial stress responses or these inflammatory responses that's really aiding with the crossing of those critical tissue barriers and viral tissue invasion in vivo. And I say this again because there's no intrinsic replication advantage early that is, those events up to and including viremia. And there's no replication advantage late in terms of these peak viral titers. We only see it at this these early seeding of tissues in vivo. And so, so I think this is really important to understand. So in conclusion, by looking at our favorite viral proteins and our favorite pathways, we've uncovered something that I think is trying to tell us something really important about the control of mitochondria, both as a kind of a source of membranes and lipids, but also potentially metabolic control in driving virus or Flavivirus, or at least Zika virus, tissue invasion in vivo. So I'm not going to go over this pathway again, but just to say that we identified a juba through interactions with Zika NS5. The juba itself is recruited to mitochondria following mitochondrial damage, where it's aiding in driving this or the early events of this critical pathway known as mitophagy. And in so doing, these viruses are actively retaining damaged mitochondria in cells. And as they do this, this is resulting in these mitochondria becoming quite visible to the cell in terms of release of nucleic acids. It may be, we haven't studied mitochondrial DNA, but this may be a reason why these viruses kind of cleave and eliminate cGAS sting signaling, as shown by Anna Fernandez-Sesma's group in Mount Sinai. But at least for the cases of this work, we've shown that it also results in mitochondrial RNA release. This is driving PKR activation, I think potentially even more so than the viral nucleic acids itself. So it's really the host that is driving activation of these pathways. This has benefits to the virus in terms of helping to fuel the integrated stress response. And I think this is fueling some of these, the ability of the virus to proliferate membranes, but it separately is able to induce inflammatory chemokine expression, and that this may have something to do with the relative amount of inflammatory monocytes in the blood, and whether or not these can either help to maybe kind of permeabilize these endothelial barriers, or whether the barriers themselves, just through chemokine expression, are somehow compromised to somehow facilitate virus seeding of these very important target tissues. And so that's kind of where we're at with the project and looking at it right now. Importantly, there's evidence of mitochondrial stress and metabolic dysregulation in the placentas from Zika infected mothers. Not more recently, but there is some evidence that this increased ISR is a contributing factor to symptomatic disease in the context of the 17D yellow fever vaccine. And importantly, it provides a mechanism for inflammation that's uncoupled now from virus replication because it's host, that is increasingly druggable due to high research interest in things like Parkinson's and Alzheimer's. So hopefully there's some avenues that we could use this as therapeutic targets. And with that, I'm sitting right in this office right now in the beautiful Rocky Mountains. And really, I've kind of mentioned the people that did most of this work, but we couldn't have done this without key collaborations from Katie Bozio, the Mouse Core, the veterinary branch, and our research technologies branch with funding from the intramural research division. Thank you very much. And thank you again for the invitation.
Thank you, Sonja. That was a great talk, going in depth into the cell and back out. Beautiful example of host-virus interactions. If you have any questions, please put them in the question and answer box. I don't think we have the chance to ask questions directly because it's a webinar format, but please don't hesitate to put in. There's one quick question at the beginning of your talk. You had the EM images and it said CM. Was that convoluted membranes? Just to clarify, there was sort of an abbreviation in there, so just to make sure. I wanted to dig a little bit deeper into the neuro connection here, and whether, this, really very detailed interaction and inactivation of that mitophagy pathway here has any implications of long-term neuro effects of people infected with Zika. I know we both know that there were sort of some emerging evidence that also in adults, maybe there might be more neuro involvement than we initially thought. And that might also lead to some potentially long-term effects in terms of memory, and which is of course frightening, but wanted to just see whether you see any connections here or whether that is just not connected.
I think this is critical because it's not only, the retention of damaged mitochondria in neurons clearly has major effects in terms of Parkinson's. And this is really, again, this crosstalk between these mitochondrial damage responses and pattern recognition receptor responses resulting in inflammation. So I think you have very clear mechanisms of neuroinflammation. But the other thing is that the mitochondria themselves have been shown to transport RNAs through, particularly in neurons where you've got these large distances that you have to cover in order to get translation in areas of at dendrites versus cell bodies, where these cells need them for function. And so if you're perturbing mitochondrial dynamics in general, I think you have the potential to perturb a lot of these very important functions. So this is the very beginning, but this is kind of exactly some of the questions we'd like to answer now.
And there's two questions and the related questions in the Q&A box. Mehdi and Sandra. Mehdi was asking whether you see any connection with SARS-CoV-2, seeing whether that would also regulate ISR regulation would also modulate tissue invasion in vivo. And Sandra wanted to know about other viruses like tick-borne virus, for example.
So yeah, so we actually found this interaction in the context of the tick-borne viruses, and so far every Flavivirus, we've got a panel of probably 8 or 9 NS5s, and they all interact. So I think this is probably more generalizable. In the sense of the coronaviruses, we are definitely looking into the coronas and the alphas. The coronas seem to completely crash the function of mitochondria, and that's associated with big changes in mitochondrial morphology. So you first get these same elongated mitochondria, followed by a massive fragmentation and loss of mitochondria. So for SARS-CoV-2, and we know from the Krogan lab, all these interactors with SARS-CoV-2 that are specifically interacting with mitochondrial proteins that control dynamics. So there's definitely some relationship. I just can't tell you how direct or how, exactly the same it is. And then the alphas are the other ones that I think are doing something very, very similar. So we're trying to look at this more broadly.
And you have a question from Frank asking whether you think that the Pkr knockout mice would exhibit less severe immunopathology, and he thinks that the Pkr might be sort of at the center of the neuropathology in flaviviruses, and what you think about it. And the second question is whether Juba is connected to interferonopathies.
Oh yeah, the interferonopathies. It could be. Certainly there's no mutations in a Juba that have been associated with inherited disorders of innate immunity, But that could be in part because of these very important functions, either in mitosis or focal adhesions is another place. So these proteins may, if they're dysregulated in terms of structure function, that may be quite severe. But we could look at that in our mice, of course, but our mice are okay. So I'm not sure how to answer that. And in terms of the interferonopathies, I think that PKR is a kinase that deserves some re-examining, because even, so I went back through some of the old Mike Diamond papers. There's a paper from Eva Harris looking at this, and Mike has always done his with a secondary knockout of RNase L, I think. So this is where these ideas are coming from, that it's completely antiviral. And I think that we just need to pass out these molecules into individual studies to kind of understand this more generally. There's another paper on bioarchives that is, or a preprint, that has the same suggestion that PKR is driving replication of both dengue and Zika. So could it be at the center of neuropathology? I'd hate to put myself at the center of anything like that, but I definitely think it needs revisiting for sure. Absolutely.
Well, thank you very much. I think we are at the top of the hour. Sonja, thank you so much for this interesting talk. Thank you everybody for coming and for participating, for all the questions. I think Sonja is going to be a little bit around and has some meetings with some of our faculty. So I'm looking forward to seeing you later, and thank you all for coming, and we see you next time.
Okay, thanks everyone.
Thanks. Bye-bye.
Bye.